So welcome to the second Build a Cell Chemical Reaction Network modeling tutorial. Today we're going to discuss compiling chemical reaction networks using Biocompiler. I'm William Poole, a graduate student at Caltech, and one of the lead developers of the Biocompiler software package. So to first start with a recap from last week, we went over what chemical reaction networks, CRNs, are. And you'll recall that these are a set of species, which I'm calling here X, but you'll see many other symbols used for them, and a set of reactions which convert multi-sets of X between each other at some rate or propensity rho sub r. And you can form a network of these reactions. When you simulate them deterministically, this gives you an ordinary differential equation given by this formula. So last week we went through an example model, which I'll go through again and use as the basis for this week's lecture. This model starts by having some external signal which can diffuse in and out of a cell. That signal cooperatively binds to a transcription factor to activate it from F0 to F1. The transcription factor can bind to a gene to form an activated gene transcription factor complex, which can then recruit a polymerase, which causes the gene to be transcribed into RNA or a transcript T. The transcript can then bind to a ribosome and form a ribosome transcript complex, which can be translated to form a protein product X. And finally, the transcript, the signal, and the protein product might be degraded via dilution because this is in a growing cell. And so this model was not too complicated, and you could imagine writing it out by hand. However, for anyone who works in biology, this is probably fairly simplistic compared to what you know is actually going on in the cell. So if we take this original model, I can show you a lot of places where we can add complexity. The first thing we might want to do is say, well, what if the transcription factor is actually being produced by a gene? And this is most likely the case. If you have something in a cell, your transcription factors also have to be produced. And so here I have to add in 6 more reactions to make that happen. I might say, well, I also know that RNA is not just degraded via dilution. There's also endonucleases or RNases that are degrading all of our RNA species. So maybe I'll add in those reactions. And I could keep going. There are many other things I could add to make this model more realistic and more complete. I could add more transcription translational machinery. So in bacteria, this might be sigma factors, which help recruit polymerases. I could add energy sources of metabolism like ATP and ATP regeneration, maybe the citric acid cycle, if I really want to go into detail there. I could add multiple occupancy of polymerases and ribosomes. So here I only allow one ribosome, say, to bind to a transcript, but in reality, more of them can bind at once. I can add background cellular processes. The ribosomes are actually being produced, and maybe those transcripts somehow interfere with everything else, and I can go on. The complexity of chemical reaction network models, when you start trying to make them accurate, grows very, very quickly. Biocompiler is a library to help you generate chemical reaction networks and deal with this complexity. As the name might suggest, it is a biochemical reaction network compiler. What is a compiler? Well, you've probably heard the word compilation from computer code, where you take some type of computer code, maybe C++, and compile it into binary machine code. And what this is really doing is you're converting a specification of what you want to have happen, the computer code, into an implementation, the binary, that a computer can understand. Biocompiler has specifications. And although previously we built CRNs by hand, we actually want to specify them and compile them. Those specifications have 3 parts. You first have mechanisms, which represent biological processes, such as transcription. You then have components, which represent biological parts, like a gene or perhaps a ribosome. We then have mixtures, which represent the context. And context comes in 2 flavors. You have biochemical context, which is saying, what type of system am I working in? Am I working in vivo, in E. coli, in yeast, in cell extract, in a minimal in vitro system, like Pure Express? And you also have modeling context. And this has to do with the assumptions you're willing to make to simplify your model. For example, I might assume that some species X is constant, and that allows me to not model it in the same way. And mixtures represent both these types of context. And finally, Biocompiler takes these specifications of mechanisms, components, and mixtures and compiles them into a chemical reaction network, CRN, which is saved as systems biology markup language, SBML, to be simulated with lots of software, as we discussed last week. Now, besides being a library, Biocompiler is also a framework. So if you're a scripter and you're trying to build models and you're a laboratory biologist or bioengineer, you're probably going to be more interested in it as a library for building models. But if you're a computer programmer or a computational biologist, Biocompiler is also a framework, and it makes it very easy for you to extend the functionality. So all of the things I was talking about, mixtures, components, and mechanisms, are objects in Biocompiler and can be subclassed. And they call each other. They have a defined relationship. Mixtures contain components. They know what's inside of them, and they also contain mechanisms. Components, in turn, know what mechanisms they need. A gene knows that it can be transcribed, and it can have either its own custom mechanism or use the mechanism that sort of is default in the mixture. And I'll talk about this more in the coding demo. When you have this whole compilation schema where mixtures call their components, which call their mechanisms, you get species and reactions, and that requires parameters. And Biocompiler has a parameter framework which allows you to use default parameters to build models quickly or specify all of your parameters in great detail. And this results in a compiled chemical reaction network. There's also one more thing called global mechanisms, which I'm not going to talk very much about today, but you can look at the example notebook I posted online, which are sort of like mechanisms, but they are owned by the mixture and they're called at the very end of compilation. These are things like degradation by dilution. So to really understand how Biocompiler works, it's helpful to go through some examples. So let's go look at this large example of the signaling network and try to break it down into the mechanisms and processes that are going on. So I might first notice there's some transport where something's going in and out of a cell. There's binding, in this case, two types of binding. One is where the signal binds to a transcription factor. One is where the transcription factor binds to a gene. There's also binding in other cases, but we'll call those other mechanisms. I then see a bunch of cases of transcription where different genes are transcribed using the same chemical reaction network motif. I see cases of translation. I see cases of dilution, and I see enzymatic degradation. And so all of these boxes represent the same process being applied to different species. And this is really the core abstraction of a mechanism. It lets us take a very complicated chemical reaction network and factor it into a bunch of processes and these smaller implementation CRNs, which are shown here on the right bottom corner. However, mechanisms are not specific CRNs necessarily. They're really black boxes. So a mechanism should take a user's input, some predefined species and parameters, and some output species and parameters, but exactly the CRN it produces can be really thought of as a black box. And I'll illustrate that with transcription. Transcription in general should take a gene and produce a transcript. Otherwise, it's not transcription. But how that works is really up to the modeler and the context and how complicated you want your model to be and what simplifications you're willing to make. For example, I might have this very simple transcription model where one gene catalytically produces a transcript, and I'm really neglecting the whole process here. I'm just talking about the idea that a gene produces a transcript. In reality, I know RNA polymerase is actually important, and maybe I'd have RNA polymerase bind to my gene to then catalyze the production of a transcript that way. In this case, I'm using a mass action chemical reaction network with 3 reactions. Or maybe I decide I want to simplify this Michaelis-Menten transcription with a Hill function, where here instead of using a mass action propensity, I'm using a Hill function propensity, but I still have my transcription rate depend upon the amount of RNA polymerase. Or maybe I want to go even more detailed and realize that lots of RNA polymerases can bind to the same gene, and that you might have some sort of multiple occupancy where there can be many polymerases on a gene that then all transcribe. All of these are valid transcription mechanisms, And they all have the same feature that they start with the gene as an input and an output is a transcript. You'll see gene to transcript, gene to transcript, gene to, in this case, multiple transcripts. And so they're all valid transcription mechanisms, but they have different CRN implementations and very different levels of complexity. Biocompiler makes it easy for you to take your specification and change which mechanism, which transcription mechanism or other mechanism you're using to model each of the processes to build lots of models of the same process and compare them. Now, components are the next key object in Biocompiler. And we'll go back to this example model we talked about earlier. Components really define biochemical functionality. So before I showed you processes, but we can also break this up into a bunch of components. Here I have my functionality on the left, transport, which is really being done by a signaling component that we know has to go from inside to outside the cell. Binding in this case is done by a chemical complex component, which are two things that sort of are complexes of multiple objects. Transcription is done by a promoter or a gene. Translation is done by a ribosome binding site, which is implicit in this case in the gene. And enzymatic degradation is done by RNases, or this endonuclease E. So you can take the same CRN and factor it in terms of both its functions, what mechanisms are happening, but also in terms of the components, what is calling that functionality. And this is how Biocompiler compiles. To give you another example of components, I want to consider this DNA construct. This is a piece of plasmid that has two promoters on it, one of which can be repressed by the TET-R repressor, and then produces a number of different fluorescent outputs. This is the type of thing synthetic biologists often build in the lab. So just to remind you, if you don't know about your SBOL glyphs, these are sort of what each of these things mean. You have a promoter, a CDS, a ribosome binding site, and a terminator. And so this component is really made up of a bunch of subcomponents. And each of these subcomponents know by design what they're supposed to do. This is why we would build this construct. The promoter is supposed to cause the construct to be transcribed. And in one case, that transcription is regulated by TET-R. The CDS and ribosome binding site together allow for translation into a protein product. Finally, you take these components and you put them inside of a mixture which defines your context. So I can take this component here that I showed you that has its mechanisms and its subcomponents and is really doing a very complicated number of things. And I can ask, how am I going to compile this? What is my context? And I do that by putting the component inside of a mixture. And I might compile this in a simple mixture. So here is a very idealized context where I'm modeling transcription and translation using that simple transcription motif I showed you earlier that has no biochemical machinery. There's no polymerases, there's no ribosomes. And you can see the compiled chemical reaction network on the bottom left. Here, the boxes represent reactions and the circles are species, which are connected to each other via reactions. And you can see that there's a bunch of reactions involving DNA and a couple involving each RNA. Or I can compile this chemical reaction network in a much more complicated context, where here I have a lot of molecular machinery like ribosomes and polymerases, and all of the complexes found by binding ribosomes to transcripts and binding polymerases to DNA. And this makes a much more complicated reaction network with many more species and reactions. And what Biocompiler makes easy is I can take my same construct, this piece of DNA, which I specify in a couple lines of code, and just by changing the context, the mixture I put it in, I can produce a very simple model or a very complicated model by really doing no more work. So let's sort of focus on this for a second, how Biocompiler allows you to explore design space. So the first thing about Biocompiler and CRN compilation in general is that it can be very modular. And what I mean by that is you can generate architectures by reusing components. So if you're a synthetic biologist or a normal biologist, you might think that, okay, we have genes with promoters and ribosome binding sites and terminators. And this is sort of a basic unit that you see in biology over and over again. And it can be, and these things can be wired together in many different ways. The simplest way I might be able to wire two of them together is to have one gene repress another. And this gives you sort of the standard repression motif you see a lot in synthetic biology. I can also have two repressors that repress each other, which makes a bistable toggle switch under the right parameters. And this is one of the seminal papers in synthetic biology, was actually building one of these in the lab. Or I can have 3 of these guys put together, and if I do this in the right way, I get oscillations, making a repressilator. Biocompiler makes it really easy to take the same DNA assembly component and wire them together in lots of different topologies using just a little bit more code. So I don't have to write out 10 more reactions for each DNA component. I can just really write out maybe 1 or 2 lines of code to wire them together. And that allows me to rapidly generate many diverse architectures, both to understand real biological systems and to design synthetic ones. Biocompiler is also expressive, which what I mean is that you can explore different circuit implementations. So before I showed you all of these idealized models, but in reality, the way we build things and the parts we choose to use or the parts biology evolved to use really matter. Biology is all about the details, and Biocompiler lets you express a lot of details. So I might take this idealized inducible repression and say, well, how might I actually build that in the lab? But one way I could build it is using dCas9 and guide RNAs, where I have now 2 different things being produced. I have a dCas9, which is a deactivated form of Cas9 that combines when accompanied with the right guide RNA to a particular part of a promoter and block transcription. Now, if I build my repressor this way, it does repress. You can see in this heat map here where I'm showing the amount of guide RNA being produced versus the amount of dCas being produced. In the far right-hand corner, the expression is going down. However, when I build something this way and simulate it in a complicated context, you also see that there's some less expected behavior. For instance, if I just produce guide RNA, my expression might actually go up of my reporter. Why is that? Well, in this model, it's because the endonucleases are being sequestered by the guide RNA and causing the degradation of the reporter transcript to decrease, which then causes an upregulation of the reporter. And this is something we can actually see in the lab. So overexpressing transcripts can actually cause something called retroactivity, which can cause an upregulation of things that normally are relying on degradation of their transcripts. And this is really important to be able to model and test how your actual implementation might affect the function of your circuit. Similarly, I could look at this transcription factor toggle switch where I have 2 different proteins that are both transcription factor repressors of each other to make this bistable toggle. And I can ask, could I build this in a different way? Well, using Biocompiler, it would be very easy to switch this to become a targeted RNase toggle switch, where here, instead of binding to a promoter and blocking transcription, each of these proteins, A and B, is actually an RNase that can selectively degrade the other transcript. And if you design this right and work in the right parameter regimes, it seems to be possible, at least according to these simulations, to build a RNase-mediated toggle switch. So Biocompiler is modular, expressive, and it helps you manage complexity. And this, it really means when you have a very complex system, you have to make assumptions and simplifications, and Biocompiler will help you test those. So if we go to this repressilator example, I showed you a very simplistic model. This is the same one from the repressilator paper by Michael Elowitz, where there's no ribosomes or polymerases. We're really just looking at a very simple model of transcription, translation, and Hill function repression. In reality, that's not how things work. If I actually look at how transcription translation happen, you know that there's multiple ribosomes that can bind to each transcript. So in this little electron microscope image, you see the little black dots are many ribosomes bound to the same transcript, which are very faint lines underneath those black dots. And this is called multiple occupancy. Using Biocompiler, I can very easily build a model of multiple occupancy for transcription or translation and see if the same repressilator topology still works. Does occupancy of ribosomes maybe interfere with how the repressilator works? And after making this model, I see that it actually doesn't. So even though I allow many ribosomes to bind to each transcript, the repressilator still oscillates. And that gives me confidence that the simplifications I made, neglecting ribosomes and their binding completely, is actually a reasonable simplification for this particular model. In some cases, it might not be. And finally, Biocompiler makes building all of these diverse circuits and architectures and exploring all of this space of possibilities very fast. So all of the examples I just showed you, I built in one day when I was writing the Biocompiler paper. And that's pretty impressive considering how big some of these models are. So this histogram is going to show you the number of species in the model in blue, which ranges from 4 for the smallest model to 180 for the biggest. The number of reactions in the model, which again ranges from 4 to over 300. The number of terms in the ODE, so if you actually wanted to write out the equation, how many separate terms would you need to do this? And this can be small. In the case of inducible repression, you could deal with this by hand pretty easily. But in the case of multiple occupancy repressilator model, there's no way you would even want to begin writing out this system of equations. It's way too big. It has over 580 terms. But the number of lines of code in Biocompiler it takes to produce this is very low, between 6 and 14. So I can very quickly and easily generate incredibly complex and sophisticated models from very simple specifications in Biocompiler code. And I'll show you in the second part of this tutorial how you actually do that coding, and it's pretty straightforward. Now, one thing though, before we move on to that, is we have to talk about parameters, because all models require parameters. And one of the hardest parts of being a synthetic or systems biology modeler is you usually don't actually know the parameter values at first. You have to wait for experiments to find your parameter values, or do a lot of literature review. And often though, you're trying to use your models to help you design your experiments. So there's sort of a chicken and the egg problem here. Well, Biocompiler helps take care of that by using this notion of successive defaulting. At the highest level, you can specify every single parameter in the model very uniquely by looking at the name of your mechanism, the part ID, this is like the name of a component, and the name of the parameter. For instance, maybe this is Michaelis-Menten transcription. Your part ID is the name of the promoter. This is one of the Anderson library promoters. And your parameter name is the binding rate. But maybe you don't actually have that parameter, and you can default to maybe just a generic binding rate for Michaelis-Menten transcription, or a generic binding rate for transcription, or even more generally, just a binding rate, a universal binding rate you're using in your simulation that's plausible until you get more complicated parameter values. And so this set of successive defaultings and some included parameter files lets you build models without specifying very many parameters at all. You're basically going to use the default parameters to get your model up and running. And then you can go in and specify detailed parameters based upon experiments or the literature. And we'll go through how to do this in the coding demo. So before I take questions, I want to give you a quick summary. Biocompiler is software written in Python that lets you compile complex chemical reaction networks from high-level specifications. Chemical reaction network compilation makes modeling more efficient, modular, and reliable. In Biocompiler, mechanisms represent biochemical processes. Components represent biochemical functionality and know about the mechanisms that they are going to call. Mixtures represent biochemical context as well as modeling context and contain both components and mechanisms. And finally, parameter defaulting will help enable the rapid construction of models before you've identified all your parameters experimentally or from the literature. A couple selected references. It's always important to put stuff in context. We have a paper out. If you want to read more about Biocompiler, please take a look and cite us if you use our software. I want to note that Biocompiler is not the first molecular compiler. This project and package is heavily influenced by the DNA strand displacement community. And I've given a bunch of references here if you want to read about some of those undertakings. There's also other compilers in synthetic biology. Biocompiler, I think, is unique in that it's trying to be a general purpose framework for biomolecular compilation, as opposed to some of these other compilers, which are really focused on specific types of systems. Biocompiler is also very similar to rule-based modeling, which is a different way of modeling that's different from chemical reaction networks, but has a lot of similarities with the abstractions made in Biocompiler. And finally, the SFOL and SVML file formats are very similar and central to this project as a way of interfacing, and so they're worth mentioning as well. So some acknowledgements. I really want to thank the members of the Biocompiler development team, Zoltan Tuza, Andre Shore, and Ayush Pandey, who've put in a huge amount of effort to make this software happen, and also thank our TAs and PIs and NSF. and the co-instructors.